atomic annihilation, the rebuilding of this great nation of ours may fall to you. That's why we at Vault-Tec have prepared these educational materials for you to better understand the 7 defining attributes that make you special. And now...
Fallout 4 is not a fantastic game. We all know that. It's mediocre at best. The biggest problem the game has, other than its optimization, is the writing.
That scientist Kellogg was supposed to track down. Virgil. We need to find him.
You're right. A rogue Institute scientist could answer all kinds of questions. Where did the memory say he was? The glowing sea? That doesn't make sense. No one goes there. Not even if they were desperate.
Seriously, Bethesda. One aspect of the plot that I never quite liked, even in Fallout 3, was the Forced Evolutionary Virus, aka the FEV. It seems to make less and less sense the further we continue with the series. I have a degree in biology. This video will be a detailed critique of why the science behind the FEV as presented in Fallout 4 is not good. I hope you stick with me until the end.
War. War never changes.
The first time the FEV becomes plot relevant is after killing Kellogg, our supervillain named after a breakfast cereal.
Okay, you made it. I'm just up ahead. My sins are standing down. Let's talk.
You murdering, kidnapping psychopath. Give me my son. Give me Sean, now!
Ain't gonna happen.
It just works.
Thank you, Todd. Sure, we can see super mutants at Trinity Tower, and we can even do a Brotherhood raid against them at Fort Strong, but neither really explain what is causing this plague of green abominations. It's the Institute, but we discover that a little bit later on in the plot. Yeah, the game has lots of wacky creatures and animals and mutants, and it turns out that quite a lot of them are the result of this FEV virus rather than the direct mutation of the wasteland. These feral ghouls, though, however, are apparently just mutated humans. I disagree with that for a number of reasons, but this video is not about feral ghouls. Right now we talk to Valentine, who brings up an old doctor capable of extracting memories from our dead serial friend.
Place in Good Neighbor called the Memory Den. Relive the past moments in your mind as clear as the day they happened. If anyone could get a dead brain to sing, it'll be Dr. Amari.
We need your help, Doctor. I need the memories from a man named Kellogg. But he's dead.
I know it's asking for a miracle, Amari, but you've pulled off the impossible before.
Please. Nick told me you're the only one who could make this work.
This dead brain had inside knowledge of the Institute, Amari. The biggest scientific secret of the Commonwealth. You need this, and so do we.
Fine. I'll take a look. But no guarantees. Do you have it with you?
Here's what I could find.
What's this? This isn't a brain. This is... Wait. That's the hippocampus. And this thing attached to it? A neural interface?
Those circuits look awfully familiar.
I'm not surprised. From what I've seen, all Institute technology has a similar architecture.
Go on, Doctor.
Mr. Valentine is an older generation synth. But Institute technology being what it is, the brain implant could fit him. But that's an incredible risk to take. We're talking about wiring something to his brain.
Don't worry about me, Amari. I'm well past the warranty date anyway.
Hey, I appreciate this, Nick.
You can thank me when we've found your son.
All right, let's get started.
Just sit down over there and keep your fingers crossed.
See you on the other side.
This technology is apparently possible in the Fallout universe. There are plenty of imaging techniques currently in use that track brain patterns. Here are a few, in no particular order. I'll even show a short table at the end. First one is positron emission tomography, aka the PET scan. This technique was developed in the 1970s and uses antimatter. No, really. It uses the antimatter equivalent of the electron, the positron. Antimatter annihilates when it contacts its normal matter version. It's a pretty straightforward principle according to Planck's concept of quantized energy. And it produces radiation at a highly distinctive wavelength and energy. Why does this matter? Well, neurons in the brain account for about a fifth of the body's energy usage at any given point. Almost all of it begins in the form of glucose, because neurons are special in the sense that they are one of the few cell types that can absorb glucose independent of insulin. It follows like this. Radiolabel an atom of oxygen using a positron-emitting isotope. Introduce the radioactive oxygen into a glucose molecule and figure out a way to put the glucose molecule into a neuron. Probably the easiest way to do this is to make sure the person being scanned only drinks a special fluid, which you are given if you ever want to get a PET scan at a hospital. You put them on a strict diet, forcing neurons to metabolize the radioactive glucose. Cells prefer not to use radioactive isotopes in metabolism, not necessarily because they're radioactive, but because they weigh more. And if the molecule weighs more, then it requires more energy to move around the cell, and will likely have less energy to give when the cell is trying to oxidize it. So after the neurons take up the glucose, we can perform the scan, which detects the distinctive radiation mentioned before. If a region of the brain is highly active, then it should glow brighter. This tells us practically nothing about the memory stored though. Yeah, big waste of time, huh? The best we can do is identify regions associated with memories. Shocker, most of them are in the hippocampus, the region of the cortex best associated with memories. It is also the most sensitive area of the brain to chemical imbalances. On a side note, yeah, most memory neurons do have their cell bodies within the hippocampus, but memory is more than just where the cell bodies are located at. Memory is the sum of all the neuronal interactions happening in the brain. A sliced portion of the hippocampus isolated from the rest of the brain is hardly useful for anything. How exactly can you remember a color, for example, if the region of the brain responsible for vision is no longer connected to the hippocampus at all? Doesn't make sense, but apparently we can do this in Fallout 4. Whatever. I guess that's fine, whatever. Let's talk about the next two types of brain imaging I'm going to mention. Nuclear magnetic imaging, aka the NMRI, we usually just call this an MRI, and functional nuclear magnetic resonance imaging, fMRI. Nuclear is usually dropped in the acronym in order to avoid scaring people, but it's nuclear in the sense that it uses hydrogen nuclei under a magnetic field. When hydrogen nuclei are exposed to certain frequencies of radiation, they enter a higher energy state. Eventually, they will return to a low energy state, but in the process, they emit a particular frequency of light in the form of a photon, which we can characterize as being part of a tissue. MRIs are static while fMRIs are dynamic, so let's continue talking about fMRIs. High-use regions of the brain should also have a high oxygenated hemoglobin turnover rate. This is because neurons require oxygen-rich blood in order to function, so obviously they're going to be consuming oxygen at a high rate compared to regions of the brain that are not directly involved in this thought process. Using fMRI, we can calculate the changing ratios of oxygenated hemoglobin to deoxygenated hemoglobin. This can help us determine what areas of the brain serve what function and purpose, and we can also have a pretty good graph of it. So fMRI sort of works here, sort of. Not really. The technology used is still magical. fMRI resolution right now is very, very low, but hey, this is in the future or in an alternative timeline, so maybe their technology is better. But our technology is very low and limited. There's also a philosophical problem here. How do we know that memory reconstruction we see is what the brain intended to create, and how do we know what the brain intended to create is actually what the person believes is happening or believes that they are seeing? If I think of the color red, for example, and you use fMRI to create an image using context from my visual cortex and my hippocampus and it turns out orange, does that mean that I was actually thinking about the color orange? Or does that shade of orange appear red within my mind's eye? Who knows? In the game, Dr. Amari implies that we are hooked up to some kind of machine. It appears that we're just walking into the memory lounger.
There's a lot of flashes. Static. I can't make sense of any of it, Doc.
That's what I was afraid of. The mnemonic impressions are encoded. It appears the Institute has one last failsafe. There's a lock on the memories in the implant.
With lots of babble of mnemonic this and mnemonic that, I can only assume by mnemonic she means the hydrostatic pressures in the ventricles of the brain. And yeah, they are important, but they don't necessarily serve for memory function. It's the neurons that do that, but the ventricles obviously do help keep the neurons healthy, and they do help support the brain. I'm not exactly sure what the writers were going for here. Maybe it's just referencing what we thought we knew of the brain back in the 1950s. But even then, we knew that the ventricles were not directly related in cognitive processes ever since, I think, the 1860s. So I'm really not sure what technobabble she's talking about. It doesn't make any sense. Also, we're not giving any kind of preparation, so I'm not sure how she can closely regulate our brain activities. And I believe that the memory lounger that Nick is in actually is unplugged, but it just works. When we get into the memories, we experience them from an out-of-body perspective, as if he's spectating his own thoughts. This literally makes no sense. All Bethesda had to do was place us in the body of Kellogg, first person. But I guess they wanted to show off this purple-black background they made. And I guess it's cool, you know, the first 1 or 2 times you play the game. Other than that, you're just wanting to finish this sequence. Because it's boring, and it's slow, and it's prone to glitches. This whole memory sequence, by the way, is continuous and in high definition. Memories simply don't work like that. Are your dreams continuous and in high definition? You may argue that it's because of the neural link chip whatever in Kellogg's brain. Stop it. Stop. Don't make that argument, please, because it actually makes even less sense. Not only would we have all the practical issues with brain imaging, we would also have to get past the firewall and encryption on the memory device, or at least the doctor would also have to be an expert computer scientist to come up with a program that interfaces with Institute technology that she has never seen before.
From what I've seen, all Institute technology has a similar architecture.
In the game, they sort of hand wave it away. Oh, well, the Institute uses similar architecture and blah, 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 blah. That's another type of bad writing. There's no reason for them to use the same architecture for every single one of their technologies. Only Kellogg was still alive, then he'd tell us. But unfortunately, he's dead. Or maybe he isn't dead?
Hey, Valentine.
Hope you got what you were looking for inside my head. I was right. I should have killed you when you were on ice.
Kellogg. Is that you?
What are you talking about?
You sounded like Kellogg just then.
Did I? Huh. Amari said there might be some mnemonic impressions left over. Anyway, I feel fine, so let's get going.
Or maybe this is just bad writing, because I don't think the game mentioned Kellogg living in Valentine's head again. So it's more of a one-off thing to shock us, rather than something meaningful to the plot. Whatever, I guess. The whole dream sequence is boring, and I hate it. After getting the brain info, we travel to the glowing sea in search of our Institute scientists. Fallout 4 has the same problem as Skyrim, where highly specific obscure locations are explicitly located on your quest marker. Kellogg didn't tell us anything about the coordinates they suspect Virgil is, but sure, it's over here in the region of the glowing sea. On our journey, we encounter one type of mutant created by the Forced Evolutionary Virus. The second most annoying enemy type in the game, only behind those stupid giant sentries. The Deathclaw. Deathclaws were hell in Fallout 3. This was a close call, sure, I admit, but only because I suck. In Fallout 4, they only scare you at high levels. Deathclaws are bipedal reptiles capable of shockingly fast quadrupedal movement. I'm not going to get too much into their biology until we finally make it to the FEV lab, but let's just say these guys were around since before the apocalypse. Their biology makes a lot more sense than the super mutant, as the game series seriously implies embryonic manipulation. So whatever. I kill a few on my journey, make a glitchy pit stop in this crater for no good reason, then finally make it to Virgil's hideout in part 2. Thanks for watching.